Hi everyone, thanks for tuning in. My name is Emily Henry. I am the non-point source coordinator for the Flathead Basin Commission out here in northwest Montana. And I will be talking about the not so glamorous world of wastewater today and some of the ways we've been developing our understanding of septic leachate pollution out here in the Flathead watershed through both modeling and these novel synthetic DNA tracer studies. Before I begin, I want to let you know I have numerous QR codes scattered throughout this presentation, so if you are interested in learning more about the content on any particular slide, feel free to scan those with your smartphone to get some more information. So to give you a bit of context, the Flathead Basin is in the northwest portion of Montana and is headwaters to the larger Columbia River Basin. The Flathead Basin Commission, or FBC as I will probably refer to it, is a non-regulatory government entity created back in the 80s by the Montana legislature with the mission of protecting the existing high water quality of the Flathead Basin. Flathead Lake and its tributaries have some of the highest water quality in the country and serve as a really important economic, ecological, and also cultural resource. So the Flathead Basin was really created to protect that. Two primary counties make up the basin. So there's Flathead County in the north and Lake County in the south. And in the southern portion of the basin is also the Flathead Reservation, which is home to the Confederated Salish and Kootenai tribes. And that is a good segue into giving a quick land acknowledgement. So all the work that Flathead Basin Commission does is on the traditional lands of the Salish, Kalispel, and Kootenai people, and we try every day to honor the path that they have shown us in caring for our remarkable natural resources, both for ourselves and for generations to come. A little bit more about the structure of FBC. So the Commission is made up of 23 seats, most of which are occupied by agencies on the federal, tribal, state, and county levels. So really, our job as a commission is just to bring all of these parties to the table to identify the most urgent challenges to natural resources in our basin and hopefully identify and implement solutions. And that brings me to the glamorous topic of this presentation, septic leachate. So for those who are not involved in this world of wastewater, when someone flushes a toilet in a home that is not connected to a wastewater treatment plant, that wastewater is likely going underground to an on-site wastewater treatment system, otherwise known as a septic system. Septic systems separate the solids in the wastewater from the liquids. That liquid is known as leachate, and that leachate is then slowly released into the surrounding soil to be treated by microbes and bacteria. Septic systems can be effective forms of wastewater treatment, but they can degrade groundwater and surface water quality if they are installed in densities that are too high or they are no longer performing optimally, which we know can occur for a number of different reasons. And we know this to be true in Montana. So there are studies dating way back to the 70s that document the impacts of septic leachate pollution on water resources. But we collectively didn't really know a lot about the scope of the issue or the spatial distribution of that. But what we do know is that this is a really complex problem with socioeconomic, political, and cultural factors to consider. So we went into this knowing that we were going to need to be really considerate of these varied perspectives. So all of that said, back in 2019, FBC created a committee of experts to further our understanding of this problem and identify possible solutions. So the first recommendation to come out of that committee was to identify the areas of the Flathead Basin at the highest risk of septic leachate pollution based on both geophysical characteristics and the age and density of existing septic systems in the ground. So getting at that spatial distribution of the problem. So to do this, the first step was to develop a physical risk model that considers 4 landscape characteristics that impact a septic system's ability to effectively treat wastewater. So the first is depth to groundwater. Obviously, shallow groundwater reduces the residence time of leachate in soil and can result in that leachate directly entering aquifers. The second is capacity of soils to treat for both nitrogen and phosphorus. So these are nutrients that are associated with septic leachate commonly and have the most influence on algae growth and water clarity. Proximity to surface water is the third. So short travel time from a septic system to a water body decreases the likelihood that soils and microbes have had sufficient time to filter contaminants. So again, reducing that residence time in the soil. And the fourth is slope. So the increased slope reduces the time that leachate is treated in the soil, again that residence time, and increases the likelihood of outflowing to the surface. So all four of these geophysical characteristics were mapped using publicly accessible data. So a model like this could really be easily duplicated in areas that already have this data published as well. And this is what the resulting physical risk map looks like. This was created for both Flathead and Lake counties, so both the northern and southern portions of the basin. And really what it's showing is how likely a septic system is to underperform in a certain area based purely on those four geophysical characteristics. We performed an additional analysis in the northern portion of the basin, so up in Flathead County, that involved looking at and factoring in existing septic systems on the landscape. So we know that age plays a really key role in how likely a septic system is to underperform. So we looked at Flathead County's septic permit database to get a sense of how old septic systems are in the county. And what we found is that over 50% are more than 20 years old. And the EPA says that the average lifespan of a septic system is 20 to 30 years. So that's a pretty significant number. We only performed this analysis in the North Basin because the septic permit data was readily available here, where that was not the case in the Southern Basin. So using this age data, we then created an age-weighted density layer that accounts for both age of systems and how close they are to one another. I mentioned earlier that the systems installed in too high of densities can overwhelm the soil's capacity for treating leachate. So that's what this layer really takes into account. I'm not going to get into the specifics of how we performed this analysis, but I would be happy to answer questions in the Hoopa app if anyone has them. So the final step in this analysis was just to combine that physical risk model, which considers those four geophysical characteristics, with that age-weighted density layer, which considers the existing septic systems already in the ground, to create this overall existing risk model. So this model identifies those areas in the basin that are the highest risk for septic system failure based on all of those things. We also just published an online interactive version of this model on our website. So you can use the QR code on the slide to check that out. And you're able to zoom into the map, move it around, turn different layers on and off, all of that. And so we're hoping that this tool will make it easier for decision makers, politicians to integrate this information into their current decision-making process when it comes to where new developments are being approved, where new permits are being issued, that kind of thing. So getting into the specifics of this study, so the second kind of phase of this project was to launch these synthetic DNA tracer studies. So going back to the idea of septic leachate being a complicated problem, we started thinking about ways in which we could definitely prove that septic leachate is entering surface water. And this is a constant struggle with non-point source pollution. Source tracking is notoriously difficult. And there are a number of different tracers that are commonly used already to do this for septic systems. But this relatively new concept of synthetic DNA as a septic tracer kind of intrigued us. And we wanted to see if this technology could possibly work in our ecoregion. So the research that our study was modeled after comes out of the University of Cornell, where the researchers sought to determine if lab synthesized DNA could allow them to infer transport pathways of septic leachate at both the local and the watershed scale. And basically, in the simplest terms, what they found is that yes, this technology does work for inferring residence time of septic leachate at both small and large scales. So we decided to conduct two very similar studies on two different lakes in our watershed, Lake Mary Ronan, kind of in the middle portion of the basin, and Whitefish Lake in the north. And these lakes were chosen for a couple of different reasons. So first, they both have homes with septic systems along the lakeshore, and Lake Mary Ronan in particular actually has a history of algal blooms. And second, both the physical risk and the existing risk models indicate high or extremely high risk areas along the shores of these lakes. So we thought maybe we'd be more likely to see septic leachate entering surface water in these particular areas. To create the tracers, we contracted with Cornell University, and at a really high level, these tracers are short, unique strands of lab-synthesized DNA that can be detected in really small quantities, basically acting as a barcode for whatever septic system they are put into. They are also encased, these DNA strands, in a biodegradable polymer, so it's actually the same material that's used for dissolvable stitches. And that allowed the DNA to persist in the environment long enough for us to go out and sample. So 8 unique tracers were developed for the 8 septic systems in the study on each of the lakes. And I am by no means an expert on these tracers or their fabrication, but I do have some information from the folks that created these that I'd be happy to share if you'd want to learn more. So once we garnered participation from homeowners and identified exactly which septic systems we were going to be flushing tracers into, we used a software called Arc Inlet to model likely groundwater flow paths from each of the systems. And this gave us an idea of where along the nearshore we should be collecting our samples for specific tracers. And again, I'm not an expert on Arc Inlet, but the QR code will take you to the research on that software if you want to learn more. And then we began the long, arduous process of sampling. So originally we had wanted to flush the tracers in both lakes back into April of 2022, but there were some delays in the fabrication process that pushed us back. So we ended up deploying tracers in May on Whitefish Lake and in June on Lake Mary Ronan. We then sampled at variable frequencies throughout the project duration, beginning with every 3 days for the first 2 months, then every 5 for the next 2 months, and then every 10 for the remainder of the study. We used Van Dorn samplers to collect subsamples along a section of shoreline. We composted all of those together and then decanted a 50 milliliter subsample that was then sent to Cornell for analysis. And they used qPCR to analyze these samples so that we were able to get relative quantities of each tracer over the course of the study period. And this is what we got for Whitefish Lake. So generally what you're seeing here is the qPCR value on the y-axis and the time on the x. And included on this graph are only the tracer qPCR values that were determined to be statistically significant and above the background threshold. Only 6 of the 8 tracers used are believed to have functioned properly. One tracer was removed due to non-specific amplification, and the other was actually not detected in either lake and believed to have been improperly fabricated. So because there are so few results, it's difficult to make statistical interpretations, but in general, what we're seeing is tracer T104, PQT, and TD2 are the worst offenders. So they're appearing in relatively high quantities really shortly after deployment. The systems that tracers PQT and TD2 were deployed into were categorized as very high to high risk by our model. So it was kind of expected that we would see those appearing, but the system using tracer T107 was categorized as high to moderate risk, so we weren't necessarily expecting this one to show the highest quantities. And these are the results for Lake Mary Ronan. So the same table setup, but overall seeing higher qPCR values of tracers in this lake than in Whitefish Lake, and persisting throughout the course of the study. And again, we're not able to make statistical interpretations here, but generally it appears that tracer T6M was one of the worst offenders, so appearing really early in the study and persisting until the very end. And the system using this tracer was categorized as moderate risk by the model, so again, not totally expected. On the other hand, the system associated with tracer TD2 was categorized as high risk by the model and appeared in relatively moderate to high concentrations consistently throughout the course of the study, so a little bit more expected there. But I think the biggest takeaways here from both lakes is that all 6 of the tracers that we believe to be functioning properly were detected in the environment at some point throughout the course of the study. Which leads us to conclude that this technology likely works for tracing septic leachates in lakes in northwest Montana where it had previously never been tested. So our proof of concept was successful, and this is just another tool in our toolbox for developing our understanding of septic leachate on surface water. We believe that with a more robust budget and timeline, we could greatly improve the results of a future study by improving our sampling methods and increasing the sampling frequency, possibly using automatic samplers to get more data and hopefully get more of those trends. We're also looking into a more in-depth site-specific interpretation of each septic system's performance in the study and the possible factors that could have been driving that, whether that's the age of the system, the soil type, system type, things like that. And finally, I would be remiss if I didn't at least mention some of the other great work that we do surrounding this issue of septic leachate. So we're working with natural scientists to identify where data gaps exist, how we might collaborate on future studies. We're working with social scientists and politicians to identify what regulations might be effective for septic pollution mitigation in our area. And we're supporting septic system owners directly by offering financial assistance to offset some of those costs of maintaining a septic system. Again, recognizing this is a complex, multifaceted issue that will likely require tackling on all of these different fronts. And a big thank you to everyone who worked on these projects, especially our volunteer monitoring crew and all of our participating homeowners. We really could not have done this without them. And with that, feel free to put any questions in the Whova app. I'd be happy to answer those. Thanks for listening.